Okay, William Neal, I'm getting a slow start here on the 18th of September, but we finally are started. I'm at the class archive page, and the first thing I'd like to say is that we've had some problems with our server over the weekend. Some of you couldn't connect. I couldn't even get my email to work on Saturday, so I didn't have time to do this, but when we start the lab session, I'm going to give you an alternate email address, so at least if somebody at a distance is desperate, or somebody local for that matter, to reach me about something, and you get a bounce back or you don't get any response on my tamu.edu email, there'll be an alternate that you can reach me by, and then if you want to send me an email with your alternate email address, we can talk to each other. I don't know, did anybody else notice their email was down on the weekend, tamu.edu? You probably just didn't check it. Yeah. So you did, Alejandro? Yeah. Saturday afternoon. And first I couldn't get my phone to work. The app on Droid wouldn't work at all, but finally they got that straight too. So anyhow, we're having a little difficulty, and we'll work around it. That's the goal. And my ultimate goal is to have a duplicate server that I can bring into service whenever the tamu.edu sites are down. Made a few additions here, one of which is the fact that I have updated the participants AVI. If you'll look down here on the info bar, and of course you can't see it when I point because I'm moving my cursor off, but I'll put it back up here. You'll notice that it's dated 9/17/12, and so that clues you that it's been updated. And I've added about 3, I think, Valdemar and who else? Cameron, maybe. And Rodney Gomez have sent me stuff, and I posted it. And there's an equivalent PowerPoint show here that you can look at if you're in a hurry, but you can't hear any. Well, one thing I want to say is that I'm not going to narrate these introductory messages anymore, because I might as well just let people read your words off text instead of listening to me talk about you, Valdemar. I think it's more effective if you talk about yourself. And so, what I want you to do, if you'd like to submit something, is send me a single PowerPoint slide with the picture or pictures that you want, and instead of writing a lot of text, just use Sound Recorder to record your message, and then send me send me your little PowerPoint show with the embedded WAV file, or send me the WAV file separately. That way it's your show instead of my interpretation of your show, and I'm not doing, I don't think, a very good job capturing what people want me to do. So what else? What else is that down here on September 13th, I not only posted the presentation I did last Thursday, which got kind of hurried at the end, and the associated PowerPoint show, but I also posted a reprint that Zoe, and it shouldn't be Elf Browning there, Zoe, that was just an artifact of my link. I saved the link by keeping the first character and then pasting in, and that way I don't have to go back and redo everything. I'll fix that. So it should be Z, or just nothing. And her colleague Allison, who isn't with us today, but this has to do with vitamin C, maybe. At least the circumstantial evidence is there that it's a scoliosis type calcium metabolism problem associated with vitamin C deficiency, and subsequent effects on skeleton and on otolith morphology, and then consequent effects on behavior, which is really a pretty neat sequence. So it's relevant to the topic I was talking about when I talked about vitamin requirements, and how in aquaculture storage of feed can result in a loss of vitamin C activity, and that vitamin E is really important to try to maintain that. I didn't say that, otoliths are the ear bones, of course, in fish, and that in teleos, that's just about the only thing in most fish that's really calcium carbonate with a protein matrix. It's called aragonite. Most of the rest of the skeleton is mainly, calcium phosphate. Yours and mine. That's one of the distinctions between vertebrates and things like crustaceans and mollusks, is that their skeletons are exoskeletons and they're, calcium carbonate, whereas our skeletons are calcium phosphate, except for the ear bones, which are calcium carbonate. Anyhow, the ear bones, as we'll learn later on, if you don't already know this, have important functions in hearing and in detecting attitude, spatial attitude, whether what's up and what's down relative to gravity. And if you have deformed ones, then you might expect, as Zoe and her colleagues did, that you'd get some abnormal behavior, and they did. So I want to get into circulation today. I'm all set up to do lab with you this afternoon. There's all the materials already posted. The handout, the doc file handout, the simulation model, the variant of ecofish that we'll use. And then I've got the black and white version of my PowerPoint slide posted, the one that I will present to the local group when we get started this afternoon, plus a raw data template, and I'm even set up to put the proceedings up once they're generated. I guess that's it. I got one other thing to tell you about, and or to ask you about locally, and that's I'll just wait and talk with the local group later because it only affects them. Let me move on now and take you into circulation, nitrogen excretion. I was thinking about the food processing material, and I had a couple of things I wanted to say more about that, but I think I'll just let it pass for now. I can't find my PowerPoint show. So we've set the stage. We've got the environmental conditions described, and we've done some simulation modeling of some of the interactions that go on of environmental conditions. We've talked about the feeding of fish and the processing of the food that fish eat, the evacuation rates that we focused on in the lab. And now it's time to distribute that stuff, and that brings us to a consideration of the circulatory system. And the function of the system is really to deal with the fact that as animal volumes increase, or plant volumes for that matter, and the volume to surface area relationship goes down, you've got a lot of distance between the outside world and the sites inside the animal or plant that are where the action is, the mitochondria. And so you got to distribute stuff, and you got to take resources to the sites of activity, and then you got to remove the waste products from those sites and get rid of them from the system. And so that's a thing that multicellular animals may have to deal with in a more serious way than single cellular animals. The bigger the animal is, the bigger the problem. And a lot of recent research on scaling of rates to sizes of systems has focused on that issue. Power laws that are so popular these days in the literature. And I'll talk a little bit about those when we deal with growth. The circulatory system of fishes is said to be closed. It's all the vertebrates have closed circulatory systems. With the exception maybe of hagfish, and there's some uncertainty still. There's new work that says maybe it isn't really, but the vertebrates basically have a system where you have the high pressure side, the arterial side of the system, and then a series of increasingly small vessels and more numerous vessels that ultimately are the capillaries that are the sites of exchange, the primary sites of exchange between the outside world and the inner tissues in an ultimate, intimate sense. And then the low pressure side of the system taking the circulatory fluid back to the heart, to the pump, and that's the venous system. So if you got capillary networks, you got closed systems. If you got sinuses and the blood just finally oozes out into the tissues and moves in the intracellular space, then you've got an open system. And a lot of the invertebrates, most of the invertebrates have open systems. And maybe the hagfish has an open system or a semi-open system. The plan of the circulatory system of fishes is familiar to you because it's the plan you learned about in human anatomy, 8th grade or whatever, physical, human, whatever the course would be that you'd take at that point in your education. And I'll show you a schematic. I'll point out that there are some important differences, that fishes have two portal systems, and I'll describe those in addition to the hepatic portal that you and I have as mammals. Fishes also have a renal portal, which is lost in embryological development in mammals. So that's my easy to understand and remember diagram of the circulatory system of a teleost, sort of generalized, obviously very schematic. Not to take the emphasis off, learning the looks of the parts as you would see them if you dissected, and focus instead on their relationship to each other and the patterns. That's what I'm trying to do here. So let's start down here with the pump, which is sort of anterior-ventral in the fish, up in the breast region. And it's sitting there. It's protected by the pectoral girdle, and it's two-chambered. There's the low-pressure atrium, or indicated by the A there, and the high-pressure ventricle indicated by V. And the heart pumps, and the blood moves anteriorly through the ventral aorta and through the gills. And from the gills, the blood is collected from the gill circulation into the aorta. The anterior branch of which is the anterior, and the posterior dorsal aorta. The anterior taking the blood to the front end of the fish, the head, and the posterior when taking it back to systemic circulation. And I've indicated just some subsets of stuff back there: the gut, the kidney, and the musculature. So, and then the blood returns, most of it, through a postcardinal vein and an anterior cardinal vein in the front, and then back through this DC, which is the duct of Cuvier, or the ductus Cuvier. Cuvier, C-U-V-I-E-R. I think I've got it spelled out in the next slide. Back to the atrium. Except for the blood that came back from the liver, which came through the hepatic veins. So what about these portal systems? Well, the first of these is the familiar one that mammals have, and that's the hepatic portal with a capillary bed on both ends and a vessel in between. And that's the definition of a portal system, is a vessel that's usually ruled a vein with capillaries on both ends. In this case, the capillaries are in the mucosa of the gut, where the blood collects the products of digestion and then carries them in that flowing blood to the liver through the hepatic portal vein. And in the liver, some of the products are processed, some are changed and modified, stored, and then some of that is mobilized and passes with the blood through the hepatic vein back to the atrium, back to the collecting organ, which is not named here, but it's the sinus venosus. I'll show you a little later. So you and I have that. You know, its role is to collect the stuff that's produced by food processing, together with a few things that are excreted in the mucosa, and those things then are delivered to the liver through this hepatic portal vein. The other portal system is the renal portal, and the upstream of the renal portal vein is the capillary network in the muscles, and the downstream capillary network is in the kidney. So there you can clearly, or you can guess probably, that what we're talking about is the byproducts of muscular metabolism that are going on in the musculature. The waste products are being transported to the kidney for processing. There's also a renal vein which takes the blood more or less back to maybe the kidney, but maybe just straight into the postcardinal. That's the reason I've got this sort of dash there. So, two portal systems. What's the upstream? Well, for the renal portal, it's the musculature of the trunk and the caudal areas. For the hepatic portal, it's the gut mucosa. What's the downstream end? Well, it's the capillary network in the liver in the case of the hepatic, and in the kidney in the case of the renal portal systems. You always give up something when you schematize things, and here what I'm giving up is the actual geometry of the relationships. And actually the dorsal aorta is about where it ought to be, but the postcardinal vein is not where it ought to be, because those of you who've done a cross-section of a fish know that both these vessels lie right next to each other in the hemal canal underneath the vertebral column. So, this thing, you have to fold it in 3 space and bring the postcardinal up there with the dorsal aorta to really make it right. But then it gets all confused when you try to understand the scheme. So I sacrifice that. So everybody sort of understands what I'm saying is that the postcardinal really belongs in the same space with the dorsal aorta. Mammals have a new vessel, the vena cava, which is formed from parts of the postcardinal and parts of other things. So fish don't have that. Fish are more primitive. The plan, as I say, is pretty much ancestral to the plan that's observed in mammals. So the heart, we'll sort of talk about some of the components now, the circulatory system, and of course the first thing to focus on, maybe the heart. I started there. I've already told you where it's located, where it is, and where it's at, and how it's protected by the pectoral girdle. Fish have small hearts relatively. A lot of fish, it's less than a couple of 0.1% of total mass. I guess being generally a little more as a proportion of total mass as the relative level of activity increases, and being more for small fish than for big ones of the same species as a proportion of mass. So it wouldn't surprise you to see that fast-swimming, highly active forms like tuna have relatively large hearts compared to more sedentary forms. 0.4% of body weight in a 1 kilogram skipjack tuna. And flying fish probably are the champs with 2.5% of their body weight as heart. And you can compare that with even a pretty, sluggish poikilotherm that's a non-fish like a frog, and it's 4% there. The heart of fishes is, as I said before, 2-chambered. And it starts out in development pretty much the same way yours and mine starts out, as a thickened region of a straight vessel. And then that vessel sort of throws an S shape as development goes on and growth occurs. And that's where it stops in the fish. And you end up with this S-shaped heart with the sort of dorsal posterior chamber, the thin wall chamber being the atrium. And then the thickened muscular chamber that's the pumping, real pumping organ being the ventricle. Looking at, well, not so much the evolution of fish hearts, I'd say the diversity of fish hearts, comparing teleos, elasmobranchs, and lungfish. Start in the middle here with the teleos heart. That's sort of looked at from above, and with the top of the slide being the head end of the fish, the anterior end, and pulling that S shape out. And so what we have is the hepatic veins and the ducts of Cuvier coming together in the sinus venosus, the sort of the collecting vessel of the heart. And then blood leaves the sinus venosus and passes into this thin-walled, translucent-walled structure called the atrium, which is renamed in higher vertebrates the auricle. There are two of them in mammals. And then through this atrioventricular valve into the ventricle, and there's where the pumping is really done, and then the blood passes into the ventral aorta, the anterior or the posterior end of which the proximal end to the heart, the near end to the heart, is called the bulbus arteriosus, and it's sort of elastic and seems to serve to sort of damp the pulsal nature of blood flow and make it more continuous. And then through the ventral aorta and through the branchial vessels to the gills and so on. The picture in the elasmobranch is pretty much the same, and that's the reason I only drew the end that's different. And that's the end up there with that chamber just downstream from the ventricle that is called a conus arteriosus in elasmobranchs, and it's actually got muscle in it. It actually is contractile, and it has these little one-way flapper valves, the little check valves to keep the blood going in the right direction. That is from bottom to top on my slide there, from posterior to anterior in reality. So lungfish anticipate the four-chambered hearts and dual circulation, as it's called, of the higher vertebrates. We see the formation of divisions between the two sides of the major chambers and also of the ventral aorta. A division formed by septa, S-E-P-T-A, well, there they are, S-E-P-T-A-E, plural. And these septae are separations, all right, but they're not complete. So there's a certain amount of mixing, and I guess the plan is that just the momentum of the flow in the blood on each side keeps them more or less separate, so that the mixing is not full. It's only partial mixing. And there's some rearrangement of these vessels back here entering the atrium. We have a new vessel formed from some old elements called the pulmonary vein that's returning oxygenated blood from the lung of lungfish. Notice it's a vein, so veins aren't distinguished by being low oxygen, or vessels that carry low oxygen blood. They're distinguished by being vessels that are on the low pressure side of the system. In this case, the most oxygen-rich blood that the lungfish has got may be the blood that's flowing through this pulmonary vein, because it's coming from the oxygenating structure, the lung. So anyhow, the blood passes from the pulmonary vein into the left side. Now this is seen from above, so that's the fish's left side on our left of the atrium. And the momentum of the flow tends to keep it pretty much on the left side as it passes into the left side of the ventricle and finally into the left side of the now divided ventral aorta, which is called a truncus arteriosus. From the truncus arteriosus, the blood flows into the dorsal aorta after passing through reduced branchial circulation or maybe even non-existent gills. The gills aren't very functional in most of the lungfishes because they're not doing much water breathing. They're mostly air breathing. And ultimately then through the dorsal aorta, the anterior aorta to the systemic circulation. Blood passes through the capillaries in these various components of the system, the muscle, the gut, and so forth. Gives up its oxygen, gathers waste, and returns that deoxygenated blood ultimately to the lung of the lungfish after being processed through the kidney and so forth. And that return is through the pulmonary artery. Trying to think how to say that or if I'm saying it correctly. Okay, so pulmonary artery through the lung. Let me pause just a second and collect my thoughts about that because thanks to my local group, I've got straightened out, I think. But if I'm wrong, then it's not their fault. It's my fault. Because all they did was nod when I talked. So, where I got off track there was after the blood passes through this left side of the truncus arteriosus and it goes out into systemic circulation and oxygen gets used up and waste get added and processed, then the blood returns through the ducts of Cuvier and these other vessels into this remnant of the sinus venosus. So now it's venous blood. It's really low oxygen venous blood and is passed into the right side of the atrium, and it tends to stay on the right side into the right side of the ventricle. It's pumped, passes to the right side of the truncus arteriosus, and there is where it goes into the pulmonary artery and into the lung, or through the lung, through the capillaries in the lung, collected. And now it's rich but venous blood that's coming back through the pulmonary vein to the left side, and so the whole cycle then repeats. So you see the idea of dual circulation, and most of you already had this idea a long time ago, but I'm kind of trying to reinforce it for you and to inform people that didn't know about it, maybe didn't have a course like this somewhere in chordate anatomy. So in the case of teleos, you got a single circulation where everything is a series. It's heart to gills to systemic tissues and then back to the heart. And the whole thing repeats, just one circuit. But in the case of lungfishes and higher vertebrates that have four-chambered hearts, for the most part, either 3 or 4. Then there's dual circulation where you've got a systemic circulation loop and a pulmonary loop. And so you can kind of think of it as a figure 8. Here comes this oxygenated blood from the lung through the left side of the heart to systemic circulation out here into the right side of the heart, through the right side of the heart into pulmonary circulation over here, back through the pulmonary vein, and so forth. So it's a dual circulatory system. This collecting vessel, notice it's moved and it's now discharging the sinus venosus into the right side of the lungfish's heart to the right auricle. And in the case of mammals, at least, that same tissue is analogous to just a patch of nervous tissue that's concentrated on the right auricle called the sinoatrial node. And it's the sinoatrial node that's the pacemaker for the heart. It's the trigger that sets the heartbeat. And of course, when you excise the heart of a fish or a frog, the heart will continue to beat because the sinoatrial node is intact and keeps firing. And the control that's central on the heart rate is inhibition, to slow down that pace through one of the cranial nerves, the vagal nerve. So that may be more than you needed to know about that, and more than I know, but I told you everything I know. The rate of pumping in a fish heart is not a whole lot less in some cases than mammalian hearts. For animals the same size, at least in tunas, you can get 100 beats per minute. We've measured that using EKGs and fish that are held in perfusion troughs or racked as they were. But, there's a concern about the artifacts of the measurement, influencing the process that you're trying to measure. There always is. You go to the doctor and you're worried about something, and naturally enough your blood pressure's up and maybe your heart rate's up. And I don't doubt that these tunas were worried about something too when they were put in that alien situation. So some of the more recent work involving telemetered fish, that are equipped with transmitting tags that transmit a signal, ultrasonic or I guess radio, that is proportionate to heart rate, that can be calibrated to heart rate. They show that the beat rates may be quite a bit less for free swimming, unstressed fish. But I think it's still accepted that pumping the beat rate is somewhere between 30 and 50 beats per minute for most fishes. More for young fish, more for tunas. It's clear that the rate increases with increasing temperature. I already told you I didn't tell you, I guess. I told you in mammals it's the sinoatrial node, but it's in the sinus venosus of fishes where the heart rate is triggered. Blood flow, I guess my numbers still, I checked something fairly recently, and this was still consistent with the most recent information. 15 to 20 milliliters of blood per kilogram of body weight per minute is typical, with extremes of 5 to 100. Elasmobranchs ranks seem to be on the upper end relative to teleos, 20 to 25 beats per minute, as opposed to 15 to 20 for fish of similar body size. Volume of blood, 1.5 to 3% of body weight. I think mammals, in the range of sizes like rats to cats is something like 6%. So that's not too far. Tunas are 7%, so tunas have blood volumes that are almost mammalian in their magnitude. And spiny dogfish is higher than ordinary teleos of the same size, about 5%. Blood pressure, 30 millimeters of mercury in a cod during the contraction of the ventricle systolic phase of the heartbeat, and as much as 80 in a salmon, and pressure drop across the gills is 40 to 50%. So on the low pressure side, about half the pressure. So you and I would be, 120 millimeters systolic and maybe 80 or 70 diastolic. Composition of the blood, two things. You got the liquid part, the plasma, and then the formed elements, the particles that are in the blood. The plasma of fish blood is an aqueous solution of of ions, inorganic ions and organic stuff, soluble proteins like albumin. And the composition is actually similar to seawater that's been diluted to about 1/3 strength. And what I've got here is a simple comparison, just a generalized comparison of the most important stuff from freshwater over here to seawater over here, and then in between the freshwater teleost. I'm not real sure it's the typical, but it's probably one where we have most data, salmonids. And a marine teleost, the barracuda. And I guess the thing to see is that fish have blood plasma concentrations of these solute materials, these things that cause decreases in freezing temperature and the associated properties, osmotic pressure. They have values that are intermediate between freshwater and seawater. So if freshwater has blood plasma that's a lot saltier than freshwater, and marine fishes tend to have blood that's not nearly as salty as the marine environment. So there's obviously a lot of regulation going on in the concentration of these solute materials, the things that are osmotically active, and the outside world. You might notice that the inorganic ions, the dominant ions, sodium and chloride, in marine sharks is actually pretty similar to freshwater teleost, more similar to freshwater teleost than to marine teleost. And yet the total activity of shark plasma is much higher. It's almost, we say isoosmotic, meaning equal total effective osmotic concentrations between the shark blood and the seawater. Measure that. One of the ways to measure it, one of the classic ways, is to measure the freezing point depression. How low, what's the temperature reduction due to the solutes in the plasma compared to the still water. And so the F here is freezing. It's not Fahrenheit, so it's delta F is the change in the freezing point measured in degrees centigrade. And for fresh water, it's negligibly below 0 degrees. I guess here our tap water, being relatively concentrated in some of these salts, has probably got a freezing point depression that's in the maybe a tenth of a degree. I'm not sure what tap water freezes at here in College Station. But this would be really good fresh water, 10 parts per million sodium and 10 parts per million chloride, or 0.01 parts per thousand. So freshwater fish have blood plasma that freezes around a half a degree below 0. Marine teleosts maybe a little more than that, about 0.8 of a degree. And seawater freezes at 35 part per thousand. Seawater freezes at 2.1 degrees below 0. And marine sharks have blood plasma that freezes at about the same temperature, despite the fact that the inorganic composition of the plasma for marine sharks is way below seawater. And of course, you know already what the answer is. It's got to do with the fact that a lot of the osmotic activity in shark blood is nitrogenous compounds that are the byproducts of nitrogen metabolism, things like urea and trimethylamine oxide or TMA is another big component. So those things are retained in the plasma and give the shark an advantage in that it doesn't have to work as hard to maintain water balance between its blood and the outside world as a teleost would, because these differences in freezing point correspond with ionic differences, and in effect the reverse of that, which is the water concentration. The water concentration of teleost blood is way higher than the water concentration of seawater. Way higher in the sense that there's going to be a lot of outward diffusion of water, or a tendency to for the water to outwardly diffuse from the marine fish blood into the seawater. Whereas the marine shark's not going to have that tendency, because the water concentration of the blood is about the same as the water concentration of the seawater. But there are some problems having to do with ion regulation. So there's still work to be done. So we'll spend quite a bit of time later on talking about ion and water regulation, or osmoregulation in the broad sense. The fact that freshwater fish are always fighting to keep out the water that's so much more concentrated in the freshwater world than in their blood plasma. And marine fish have just the opposite struggle, marine teleost, but not marine sharks. That is, keeping their water inside instead of letting it escape to try to dilute the water that's in the world's ocean. So that's the thumbnail sketch of the plasma. What about the formed elements, or the particles that are in the blood? And so I'm going to talk about red blood cells and white blood cells now. Red blood cells are erythrocytes. They're actually not erythrocytes. They're true cells in the case of fish, and that's the first thing to pay attention to, I guess, is in the case of teleosts, the active red blood cells, the mature red blood cells, the ones that are circulating and carrying the oxygen and carrying the carbon dioxide and other things, are actually nucleated. Whereas in our blood, yours and mine, the mature cells are no longer cells; they're sites. They're without nuclei. So fish have nucleated red blood cells, and these red blood cells make up a fairly large proportion of the volume, 20 to 50%. The hematocrit, or the packed red blood cell volume, compared to what's left, the plasma, approaches 50% in some fish. I've always wondered about the value of hematocrit and its meaning, because I had a little demonstration from a colleague, Dr. Don Stevens, who spent a semester here with us on a sabbatical once, and he showed me a movie with some rainbow trout in a shallow tank with a glass bottom, and the camera looked up from underneath the tank at the fish, and these fish had their abdominal walls removed and replaced with plastic, so that you could actually see their internal organs as they were swimming around. And when the fish were panicked by being suddenly approached with a net, you could actually see the spleens of those fish contract. And the spleen is a storage organ. It stores red blood cells when they're... what I learned from that experience was, I guess, what Don Stevens already knew, and that is that fish, and humans too, are, just recruiting into systemic circulation the oxygen transport capacity that they need at any particular time. And if they're having to do a lot of work, or if they're in a panic state, then they have a lot of their red blood cells are in circulation. And if they're not, they just sort of collect them and hold them in reserve in their spleens. And so when you measure hematocrit, and you grab that fish, and the fish is out of the tank and it's struggling, and you draw that syringe full of blood, or cut its caudal end off and drain it, I suspect that maybe the value of the hematocrit that you're getting is not maybe the normal swimming around hematocrit. A lot of things are like that. We have to worry about the measurements that are made being that they're pretty intrusive and traumatic, maybe affecting the results. Red blood cells are not absolutely essential. The cyanithid ice fishes of Antarctica have no red blood cells. They have no hemoglobin. They got colorless blood. Leptocephaly, the larval form of eels, are basically two-dimensional sort of willow leaf shaped structures, and so they don't maybe need a lot of oxygen transport capability that would be afforded by red blood cells, and they get by without it. That's a developmental thing. They gain red blood cells and hemoglobin as they develop. And the most surprising story I've ever heard is this story that I think a guy named Holton and Dave Randall at UBC spent a lot of effort tracking down this story to be sure that it was legitimate, and I think it all checked out. But this fellow named Schlichter in Germany reported a case involving a age 1 carp, not a young of the year carp, but a carp that had been through a whole growing season and a winter that looked to be genetically without hemoglobin and without red blood cells. So what you got to wonder is, how could you survive to be 1 year old without hemoglobin? Holton went on to do some experiments with rainbow trout, and what he did was take rainbow trout and deactivate the oxygen transport capability by poisoning the hemoglobin with carbon monoxide. And that's what, carbon monoxide irreversibly binds with the hemoglobin's binding site, so you can't carry oxygen. That's the way carbon monoxide poisons. Anyhow, what Holton found was, Holton and Randall, that rainbow trout could survive carbon monoxide poisoning if they were at a low enough temperature, below 5 degrees. But if you raise the temperature even to 10 degrees, then they died basically of suffocation or hypoxia. So what's the story here? Well, I guess the implication of the story is that enough oxygen can be transported in solution, provided, of course, that the demand for that oxygen is very low, that there's not a lot of need for it. In other words, the animal can survive as long as metabolism is going on at such a slow pace that the oxygen transported in solution is adequate to meet the demand. But most fish, I think it's safe to say that most fish under most conditions would not be able to handle no hemoglobin. I'll have more to say when we talk about respiration about the actual role of hemoglobin in oxygen transport and carbon dioxide transport. The white blood cells that fish have are similar to the ones that we have some of the same names. I don't know if I even name them, but I think they got monocytes and polymorphonuclear leukocytes, and they got all those things. The functions that they have are about the same as in mammals. They got some ones that do some scavenging, going around and removing debris from the blood. They produce antibodies. They're important thrombocytes and blood clotting. Blood formation, pretty generic stuff. Plasma is sort of the blood plasma. It's sort of the sol form of cytoplasm that leaks out of the cells and is collected in the sinuses. And red blood cells are produced by the spleen in fishes, and they're also decomposed there when they wear out. They're also produced in cellular marrow-like areas of the larger skull bones. And you and me, the long limb bones and the marrow areas of those are important in production of red blood cells. But fish obviously don't have that going for them, so they use the larger skull bones. White blood cells are produced in the spleen, the kidney, and the submucosa of the gut. Fish have a lymphatic system, which is sort of this parallel ghost system of circulation with the veins and arteries. The lymphatic system is derived from the venous part of the circulatory system, and it serves to collect the extracellular tissue fluids that sort of escape and put them back into blood circulation through the cardinal veins. So I want to move on and now talk about nitrogen excretion. And the text, if you've got it and are reading along, chapter 24 of Bond's second edition is the one that covers that. Protein breakdown products or metabolites are a problem for fish in that the nitrogenous excretory products tend to be toxic, or if they're more complex compounds that aren't so toxic, they take energy to form. Fish mostly excrete ammonia, but some fishes also, and I already mentioned that elasmobranchs produce urea and some other products. The breakdown products of carbohydrates and fats tend to be relatively, as I say, they're trouble-free. The only metabolites that are there are carbon dioxide and water. And carbon dioxide can be a problem under low pH conditions, but typically it won't be. So I'm going to talk about, spend some time talking about nitrogenous wastes, and I'll refer you first to a classic experiment done by a fellow named Homer Smith back in the late 1920s. And it was Smith's experiment that unequivocally demonstrated how nitrogenous waste pass out of fishes. What he did was take a fish, I think goldfish, and divide the head end, or separate the head and tail ends of the fish with a partition in the aquarium, a flexible rubber membrane. Put the fish through the hole so that the head end and the gill end, particularly, of the fish are on one side, and the vent, the anus and the urogenital opening, was on the other side. And then he measured what accumulated in the water. And what he found was that most of the nitrogenous waste products that accumulated on the head end of the fish. And he actually leaped to the conclusion that it was from the gills. That was pretty logical, but it took several years before people actually demonstrated the absolute, that it didn't come out of the mouth or the mucous lining of the mouth or the eyes or something else. But it's, sure enough, it's out of the gills. And it's mainly ammonia, but it's also some urea, amine or amine oxide derivatives. And then on the tail end of the fish, coming out from the kidney and the intestine, were creatine and creatinine and uric acid. So when I ask you on the exam to tell me about Homer Smith and why his work was important and what he found, how he did it, you're going to tell me, right, that he took a fish and he put it into an aquarium and he separated the head and tail ends with a flexible membrane and measured the accumulated nitrogenous materials. And what did he find? He found that the majority of those compounds appeared on the head end, presumably coming from the gills. And the great majority of what was there was ammonia. So two things, mostly from the gills and mostly ammonia. Ammonia from the gills was 6 to 10 times the total of all the nitrogenous compounds that came from the vent, or the posterior end. So there's the conclusions. Fish excrete 60 to 90% of their nitrogenous waste from the gills, and most of the nitrogenous waste excreted by fish is ammonia. In fishes, kidneys are mainly organs of salt and water balance. Presumably the kidneys developed evolutionarily as water balance organs and ion balance organs, and developed this waste processing role secondarily. There is some stuff that goes on that I don't even know if I have in my notes. Probably not. But the kidneys actually do have a role in processing nitrogenous waste from the tissues, that renal portal system I talked about. And they extract, it's the alpha amino ammonia from the breakdown of amino acids, and that's transported then in the blood to the gill where most of the actual excretion occurs. So it's carried away from the kidney in the blood to the gill where the ammonia is excreted instead of going out as urine. Advantages of ammonia for animals that live in the water and breathe water. Advantages over urea and uric acid are that you don't have to invest any extra energy in making it, because it's the natural deamination of the amino acid, the breaking off of that NH2 group, and you've got NH3. That's it. Ammonia has a small molecular weight. Just ammonia, it's just nitrogen and 3 hydrogens, or 4 hydrogens with a charge. And so it's highly soluble and moves easily through membranes, and so that makes it easy to eliminate by diffusion across the epithelium of the gill without a lot of extra water being passed out with it, which is always a cost of urine formation, is the water that you have to use to dilute the nitrogenous compounds to the point where they'll be in solution. And that's not a big problem if you live with lots of water availability, but if you live in situations where there isn't much water, it can be a big problem. In freshwater fish, there may be some advantage in terms of exchange diffusion between the ammonium ion going out and the sodium ion being recruited, because the fish is always going to be wanting to pick up as much sodium as possible from the water, because it wants to take rare ions up from the water if it's a freshwater fish. Keep them in its blood. Remember, freshwater fish have the problem of resisting influxes of water from a more watery outside world, and resisting the loss of these rare and hard to get ions that had to be scrubbed from the water to start with. And so it might help, if you could get rid of one waste product in return for recruiting something that you need. But that doesn't do much to explain what happens in marine fish. So I'm not real sure I like that argument. Big disadvantage of ammonia excretion is its toxicity. And that's not so much of a disadvantage if there's plenty of water to dilute the ammonia in, as there most often is in a natural situation where you've got low biomass densities and low reduction of ammonia per unit volume. But in situations where water's in relatively short supply, it can be a real problem. And one natural situation is an estivating lungfish, which is in a little burrow in the mud, a little cavity, and it's lined that with mucus, and it's excreting things into that mucus layer, and pretty soon, if it were ammonia, the ammonia would reach lethal levels. So estivating lungfish have to go ahead and do the extra work that mammals and birds do and convert that to something that's not as toxic, so they convert it to urea. Elasmobranchs, the sharks that produce eggs in hard cases. You've maybe seen the skate egg cases that sometimes are found on the beach. They do the same thing. Embryologically, the embryos that are developing are converting ammonia to urea so that they don't poison their limited embryonic fluids, I guess you could say, inside the case, either inside the mother shark or in the case that's outside. Some of the sharks develop these eggs inside. Others lay the eggs and they hatch outside. So they protect the young from ammonia toxicity during a long period of intrauterine development or egg incubation. And another place that is maybe more familiar to most of us here in the room, at least, maybe not all of us, but is intensive aquaculture, where you got a lot of biomass and a lot of metabolic activity going on, a lot of protein being consumed, and a lot of nitrogen being excreted. And one of the big problems that you have in intensive aquaculture is ammonia toxicity. The way we deal with it is mainly by a method called biofiltration, which is community ecology in a very organized sense, where what you do is provide surfaces that can be colonized by appropriate types of bacteria that specialize in metabolism of ammonia. And one genus that's well known is Nitrosomonas, which converts ammonia to nitrite, NO2 minus, and nitrite itself can be toxic under certain conditions, in freshwater particularly. Causes a condition called brown blood disease, and that's sometimes a real problem in catfish culture. Not so much in marine fish culture or estuarine fish culture, because the chloride ion tends to protect against the brown blood disease problem. Anyhow, nitrite is not anything like the culprit that ammonia is. And even if it were, it can be converted on down to or oxidized to nitrate by another oxygen addition. So you get NO3 by this group of bacteria that is sort of represented by Nitrobacter. So you got Nitrosomonas that processes the ammonia, converts it to nitrite, and then Nitrobacter takes over and converts the nitrite into nitrate. And nitrate is almost non-toxic. It has to be very high levels, although there are situations where, and there are organisms that are so sensitive. It's a combination. High concentrations in sensitive organisms that you can have trouble with nitrate. And I'm thinking about Dr. Philip Lee, and who cultured squid and what else? Squid and cuttlefish, and these things are super sensitive. And he was actually converting the nitrate to nitrogen gas and getting it out of the system. You could either do that or you could replace the water. But the water he had to use was very, very pure and had to be hauled from way offshore, so it was pretty expensive to do that. In transport of fish, fish eggs and larvae, and obviously when you are transporting fish, you'd like not to have to transport a lot of water, because that's where the weight is. So one thing that folks do is try to buffer the system to make ammonia less toxic than it would otherwise be, and that means buffering it, buffering it down, right? Below 7 maybe. Ray's looking at me and he's thinking too, but he hadn't thought this thought in a long time. I think that's the risk, you know. At high pHs, most of the ammonia tends to be in the ammonia, the so-called unionized form, and that's the toxic form. So if you can buffer in the other direction, maybe you can keep it in the NO4OH form or something that's less. Pardon?
NH4 plus.
NH4 plus form. Okay. So buffering anyhow is a way to try to control. Well, I go ahead to say I didn't have to guess there, right? See, buffering to maintain the optimum pH. Toxic forms of ammonia are the unionized forms NH4OH and NH3, which predominate at high pH. Ionized ammonia is NH4 plus, and that's relatively innocuous. The proportion of total ammonia in the unionized form is a function of pH and temperature, and I've got a little Stella model that basically returns the values that are in the table by Emerson et al. In 1975. Sort of simulates the processes of association-dissociation. And I don't have that posted, but I will post it so that you can have it if you want it. And we ran over, I guess, didn't even notice what time it was, and people were very patient with me today. Got to get out of that habit. Julie's going to let me know next time. Right? Or Zoe or somebody. Chris looking at the clock, checking me. I got a patient bunch of folks here today, so I appreciate that. We will do lab this afternoon. See most of you there, I'm sure.